How do you prepare a slide of cells to observe under a microscope? This usually comes up as a 3-mark question, and it's actually quite a common question in both foundation and higher. So again, I would advise that you memorize this. You take the onion skin, peel thin layer, or you can take cheek cells and use a cotton swab, or you could use pond water, where you can use a dropper like this to get the water. Whatever you've taken, we call that a specimen, and we put at the center of a clean slide. We use filter paper, and then what we do is we add our iodine solution, add a couple of drops, and that stains it. Then you put a cover slip on top. When you put the cover slip on top, sometimes what happens is you get a bit of excess liquid, excess starch solution, or whatever stain that you used. Now, what you do there is you take a small amount of tissue paper, and it just absorbs that excess liquid. Otherwise, it can ruin what you're looking at. And alongside that, you need to know how to use an actual microscope. And this is, again, a common question. How would you set up and use a microscope to observe cells? So you place the microscope on a flat surface, plug it in, especially if you're using a light microscope. Now, that might seem like a trivial point, but I've seen it in mark schemes. It is a core part of using it. If you don't plug it in, it doesn't work. You've got some that have got mirrors. So with those ones, you would just point the mirror in the direction of the sun. That will get you the mark 2. Eyepiece, and adjust the coarse focus. So you've got the eyepiece lens that you're looking through, which usually has a magnification of ×10, and you adjust the coarse focus. Now that coarse focus causes it to move up and down. Now initially you would take the objective lens so it's touching the stage. Not touching, but as close to the stage as possible without it touching the slide that you just made based on the previous slide. Then you start turning the coarse focus so it is really, really close. Then you slowly bring it up till you can kind of see the image. The fine focus brings up the objective pieces more slowly than the coarse focus, and it helps you get that really clear image. And then you would draw whatever you can see. So if you can see multiple cells, you're going to draw them all to the best of your ability. And then you would do the same thing. Initially you do it at ×10, then you try it at ×40, ×100, depending on how good the microscope is. But again, you need to know those steps. Now there are some risks. And again, if you've got a core practical exam question, you may get questions within the evaluation, or you may get questions linked to hazards. It's a very common question, so you need to make sure you know that iodine solution, if you use that as your stain, it can be toxic. It can irritate the skin, the eyes, and it can stain clothes. So we only use small amounts. We can wear gloves, or if you're using a dropper anyway, it is not massively a problem if you don't have gloves on. And if it spills, you clean it immediately. If it touches your skin, you wash it off immediately. Then, if, for example, using pond water, you can get contamination with bacteria, and it's important then to wash hands after handling. Dispose of samples properly. Now, describe how this is an actual past paper question. I think it was a 4-mark question. Describe how this equipment could be used. Also, just before I continue, this is the kind of question that you would find at the end of a foundation paper and at the beginning of a higher paper. So describe how this equipment could be used to prepare a slide of onion cells to view under a microscope. So again, this is the kind of answer. Whenever you're doing past paper questions, if you come across an answer like this, it is always beneficial to just memorize it. Because look, it's giving you every single step. Pretty much almost the same as what we just went through. Add a drop of water to the microscope slide. Okay, we know that from what we just learned. Place a thin layer of tissue on the slide. The tissue comes from here, from the onions. Stain the tissue with a couple of drops of iodine solution. So again, we've just covered that. And then finally, place the cover slip on top. And we've got our 4 marks done. Describe how this equipment, so if you now compare that to the mark scheme, because that's what we've already learned, you know, that's 4 marks out of 4, or 3 marks out of 3. What we did cover in what we were going through stuff, we didn't cover the forceps, because you can use forceps to pick up the thin layer of onion cells. But we've covered placing onion cells on the microscope slide, adding a drop of stain, placing a cover slip on top, and lowering the cover slip down slowly at an angle. Now, the student squashed the root tip on the microscope slide to spread out the cells. The slide was placed on the stage of a microscope. Describe how to use the microscope to obtain a clear image of the cells. So this question is just saying how to use a microscope. Again, it's just worth memorizing these kind of questions. Place the slide on the stage and use the lowest power objective lens. Turn the coarse focus wheel to bring the image into focus. Increase the power of the objective lens to increase magnification. And done. That's your 3 marks. So remember what we said before? Well, this case says 2 marks. Saying that switching the lamp on, you get a mark. Starting with the lowest objective lens, then using the fine focusing wheel or the coarse focusing wheel to obtain a clear image. Don't just wing it.